Hello student, welcome to EPG Pathsala. I am Dr. Ajeet Jaiswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about the modules called as Applied Human Genetics under paper Physical or Biological Anthropology. The learning objective of this particular modules are as follows. In this modules we are trying to find out the answer of some of the important questions like what are the scope of human genetics? What could be the role of genetic trait in population variation? How could the inter-population variation leads to varied prognosis of complex disorder? What are the major area of healthcare that human genetics can be applied for? What would be the effect of technological advancement in this subject matter, especially in case of forensic application? What would be the source of genetic information from archaeological remains? Now let me give you a introductory information about this particular modules. Genetics has become the important part of our daily life. As we know that genetics is about the inheritance of gene from parent generation to the next generation, and also it is associated with expression of phenotype from genotype. This basic concept of genetics can be applied to a number of basic or applied researches as such, or it can be controlled or modified to get the desired result. So, in fact, the application of human genetics has become more broader in the post-genomic era. Applied genetic researches are usually carried out to dig upon practical problem or to implement for the better human society, such as genetic predisposition of certain diseases, solving the human evolutionary queries, forensic application, etc. In this module, some of the important application of the same is discussed. Population variation. The concept of genetics has been applied in exploring the human genetic variation and hence depict the population history or evolutionary framework of biological species. Individuals in a Mendelian population share a common gene pool that can be traced back to their ancestor. Every population has its own genetic structure, which can be defined as the distribution of allele and genotypes in the population. Statistical inferences plays a crucial role in reconstructing the evolutionary framework. The basic principle in this approach lies in the concept that evolution could be constructed by analyzing the multilocal genotype on a chromosome observed in populations and their inheritance within families. Population variation and tools for study. Genetic variation in both inter and intra-populations are studied through certain selected genetic marker. Genetic marker can be either serological or biochemical traits. In the post-human genomic project, genomic markers are more considered as a more reliable tool for studying human genetic variation. There are certain basic characteristics to be considered for genetic marker, such as specific locus, polymorphic and easily genotyped. It is through this genetic variation that one's population structure can be determined, and at the same time inter or intra-population variation can also be evaluated through certain statistical models. Phylogenetic relationship of the population can also be determined based on the genetic similarities and differences. Consequently, exploring human genetic variation has become one of the important applications of human genetics, and thereby trying to give an insight on the human evolution and the earliest migration. Moreover, the knowledge of human genetic variations can also be applied in understanding the role of genetic variation in the development and progression of complex disorder all over the world population. It is assumed that the pattern of genetic variation they set light on recombination, demography, admixture and evolutionary selection in the human population, and thereby projecting the underlying genetic manifestation of the disease varied population wise. Further, population heterogeneity may lead to spurious associations while examining the genetic role in disease progression. It is hereby noteworthy to mention that good knowledge of population genetics or population structure is much needed in understanding the genetic mechanism of disease progression, which may differ from population to population. Medicine and healthcare. Genetic technology is now an important part of medicine, and its impact will continue to grow as information from genomics is analyzed and applied to the diagnosis and treatment of human diseases. Genetics has a very important role in medical aspect, thereby promoting healthcare. Hence, an independent discipline has been emerged as medical genetics. With the concept of one gene, one enzyme, the importance of gene in development of Mendelian disorder or classical single gene disorder cannot be ignored, as disease can result from altered proteins or too little or too much of a protein, or proteins made at the wrong place or time. Moreover, various complex disorders are also considered as the consequence of the interaction of gene and environment. Even the inherited ones are sensitive. Some of the applications of genetics in medicine and healthcare could be analyzed as such. Genetic screening and counseling. Screening of various genetic disorders, both single gene and complex disorder, is very much essential for the promotion of common health care. There are a number of inherited disorders which are lethal or sublethal. Screening of such lethal or sublethal disorder is one of the important applications of human genetics. It is expected that millions of people worldwide are affected to such genetic disorder, with over 10,000 monogenic inherited disorder, and every newborn has a 3% chance of having a genetic disorder, underscoring the need for tests that accurately diagnose heritable diseases at all stages of life. These monogenic disorders may be autosomal or sex-linked inheritance in either dominant or recessive pattern. Dominant inheritance will result into the expression of phenotype or disorder in question even in presence of only one copies of mutant allele, while for recessive trait, two copies of mutant alleles are needed to express the phenotypic traits. The concern of human geneticists in this regard lies in the identification of early prognosis of such lethal or subgenetic traits, especially among the newborn, and hence remedial measures can be planned through counseling. Let's try to see the reasons to seek genetic counseling. First one is family history of abnormal chromosome. Next, elevated risk of single gene disorder. Next, family history of multifactorial disorder. And next one is family history of familial complex disorder. For this, the genetic counseling sessions are like finding out family history, pedigree construction, information provided on specific disorder or modes of inheritance, test to identify at risk familial members, testing arrange discussion of result, links to support groups, appropriate services, and follow up contact. Genetic counseling. Genetic counseling is the process by which the patient or relative at risk of an inherited disorder advise of the consequences and nature of the disorder, the probability of developing or transmitting it, and the option open to them in management and family planning. This complete complex process can be separated into diagnostic, that is the actual estimation of risk, and supportive aspect, that is related to supporting. The person who is giving advice is the counselor, and the person who is seeking is counselee. Counselors need special attention in case of monogenic recessive disorder such as cystic fibrosis, phenylketonuria, etc., as the normal couple has the probability of giving birth to affected children in the condition that both are carrier of disorders. Early prognosis of such disorder is very much essential for the babies. Beside the monogenic inherited disorder, the counselor also deals with the complex disorder and the probable interaction of environment and gene based on the incident of a family history of the disorder. Though the counselors talk about the causes and consequences of genetic disorder, it is the counselee who has to take the final decision in this matter. A counselor cannot insist the counselee to opt or decide in favor of him. Genetic epidemiology. The genetic epidemiological study or epidemiological study is one of the another important application of genetics in the field of health science. It shows the relationship between population genetic structure, that is interpopulation variations, and also genetic epidemiology. So there is a continuous chain of relational flowchart can be seen in the case between population genetics and genetic epidemiology related to genetic epidemiology. According to John Hopkins Bloomberg School of Public Health, genetic epidemiology is the study of how genetic factors contribute to health and disease in families and population, and how genes interplay with environmental factors. So it can be inferred that it is the study of interaction of genes and environment on health and disease risk in population, which is very resembles with anthropological approach of nature-nurture interaction. Genetic epidemiologists attempt to identify the many components of risk attributable to genes, environment, and interaction between these two factors in the progression and manifestation of various disorders, especially of the complex one. In the post-human genome era, technological advancements such as implementation of genome-wide associated studies, sequencing, etc. have brought a new dimension in the biomedical research with identification of various disease susceptibility loci and interaction among each other in the phenotypic expression of the trait under concern. Moreover, the epigenetic approach also contributed understanding to the role of gene and its associated factor in the varied disease expression. Genetically, no two individuals are identified. Similarly, one Mendelian population differs from each other in terms of its genetic structure. Individuals in a population are believed to share a common gene pool, and hence disease susceptibility loci are more or less common among the individuals of the same population. Here, the importance of population genetic variation lies in analyzing the disease susceptibility loci in different populations, and so forth. Different disease management programs can be taken up. The interaction of human genetic variation in epidemiological approach is well evident through demographic parameter, as the changing demographic aspect may transform the population's gene pool, which in turn could switch on or off the clinically important risk allele or gene and thereby affecting the community health with complex disorder. Let's discuss about metagenomics. The metagenomic studies are also a part of genetic epidemiological approach. The role of microbes and its genome in understanding the overall human health cannot be ruled out. Metagenomics, it is the study of genetic material recovered directly from environmental samples, has emerged as a powerful tool that can be used to analyze microbial communities regardless of the ability of a member organism to be cultured in the laboratory. Metagenomics could also unlock the massive uncultured microbial diversity present in the environment to provide new molecules for therapeutic and biotechnological application. The application of net genomics can be more clearly understood in the context of human nutritional studies. Nutrients are important basic needs for human body, even for body functioning. But the problem lies that at the present, the world is witnessing dual phase of nutrition, that is undernutrition and overnutrition, which both are harmful to the human health. Absorption and digestion of nutrients have biological implications. The microbes present in the intestinal tract have a major role in the overall processes. The interaction of microbes and its genomic content need to assess to address the human nutritional problem. Forensic genetics. Personal identification is one of the important objects in forensic investigation on the ground of a medico-legal context. Identification can be done through different biological traits, but genetic traits give more precise information in this aspect. It is already established fact that no two individuals are completely identical, and this inter-individual variation is more apparent when genetic analysis is made. It may be case of paternity disputes or identification of victims or culprits involved in any forensic related issues could be solved through genetic identification. The application of genetics in the medico-legal context for identification of person involved in it is referred as forensic genetics. In earlier classical genetic traits have been employed for the same, but in the post-genomic era, DNA profiling known as DNA fingerprinting have been used quite extensively. DNA fingerprinting, also popularly known as DNA profiling, is a specialized method to analyze, compare and identify multi-local DNA binding patterns that are specific to an individual by exposing a sample of the person's DNA to molecular probe and various analytical techniques. It was first discovered by Professor Alex Jeffrey in the year 1984. Biological sample can be collected from different sources such as buccal swab, blood, saliva, semen, any cellular material, and further analysis like DNA extraction, amplifications and genotyping are done. Through DNA profiling, victim identifications, even in natural disaster tragedies such as plane crash, man-made disasters such as terrorist attack or genocides can be done. It is reported that DNA profiling has been rarely used before September 11, 2001, but after the incident of World Trade Center fall, it becomes an important tool for personal identification even in the case of missing people. The well-published case of Congress veteran leader N.D. Tiwari can be cited over here. Prior to the arrival of DNA profiling, identification can be assessed from serological traits, blood group, etc. As an example, in the case of paternity disputes, the probable fathers of an illegitimate child can be found out by comparing the blood group of mother, child, and suspected father. It is based on the inheritance of blood group, as O blood group parents cannot have A or B blood group children. Archaeogenetics. Human evolution itself is a mystery. There are lots of obscurities that need to be explored to have an overall illustration of human evolution. Different stages have been constructed based on the fossil evidence, though there are missing links which hasn't been rectified yet. In fact, the relationship or interaction between the extinct and the extant hominids has not been clearly depicted. The emergence of population genetics in the evolutionary biology, particularly in the human evolutionary studies, led to the development of new discipline, archaeogenetics, with which explain the human past through molecular population genetics. It is defined as the study of the past by use of technique of molecular genetics. This discipline targets to collect genetic information from the fossil hominids and make it compared with those of the present living human population group, and tries to decipher the genetic relationship of extinct and the extant human population and overall the probable relations with the environmental factor through statistical models. Different studies have been come up from Europe as a number of archaeological sites are discovered in the 19th and 20th century. The issues related with the human early demographic history are the main concern of the archaeogenetics. Besides this, it also concerned with the archaic pathogens that affect the health of extinct humans through historical epidemics. The significance of archaeogenetics lies in the concept that it can shed light on the origin and geographical spread of prehistoric languages and also assist archaeologists in answering questions regarding the influence of population growth in the archaeological record. As an example, mitochondrial DNA analysis of modern population of South Asia, East Asia and Oceanic reveal that there was larger population expansion before the advent of microlith technology. Through a molecular clock analysis, this expansion was dated to about 38 to 28 kiloannus, which is a unit of time equivalent to 1000 years, while microlith technology is expected to started from 35 to 30 kiloannus ago. Now let me give you the conclusive information in the form of conclusion about this module. The application of human genetics is increased beyond the horizon. Biotechnological application of the same improve the quality of human life by providing genetically modified foodstuff. It also formulates the substratum for new diagnostic test or synthesis of a customized protein for treating certain disease or production of a vaccine in medical arena. it enhances the concept of community health and also assist in the population specific drug designing pharmacogenesis based on the concept that every population have unique gene pool. The important points that we have learned in these modules, let's discuss in this point in the form of a summary. The application of human genetics in the present day world is vast. Gene pool of a population can be traced back to their ancestors. Evolution could be constructed by analyzing the multi-locus genotype on a chromosome observed in populations and their inheritance within families. Gene markers can be either serological or biological traits. Genetic markers should be locus-specific, polymorphic, and easily genotyped. Pattern of genetic variation project the underlying genetic manifestations of the disease. Complex disorders are now considered as the consequences of the interaction of gene and environment. Screening of lethal or sublethal disorder is one of the important applications of human genetics. Genetic counseling is the process by which the patient or the relative at risk of an inherited disorder are advised of the consequences and nature of the disorder, the probability of developing or transmitting it, and the option open to them in management and family planning. Genetic epidemiologists attempt to identify the many components of a risk attributable to genes, environment, and interactions between these two factors in the progression and manifestation of various disorders, especially of the complex one. Absorption and digestion of nutrition contribute to the manifestation of nutritional status of the individual. The microbes present in the intestinal tract have a major role in the nutritional health. DNA profiling is latest technique for identification of person in the context of medico-legal issues. Thank you.